Hello students, I'm Imkong Tulnapungan from Department of Anthropology, University of Delhi. Today we will be discussing on the module Molecular Phylogeny of Living Primates from the paper Human Origin and Evolution. Well, the learning objectives of the module are to understand the molecular phylogeny of living primates, to understand the indirect methods to study molecular phylogeny, to understand the direct methods to study molecular phylogeny. Firstly, let us discuss briefly about evolution. Evolution is a complex subject encompassing the history of life and its governing processes. Our understanding of evolution is based on interpretation of patterns of diversity, ecology, behavior, visible morphology and invisible molecular structure that have changed and continue to change through time. The widely varying organic forms that surround us are a product of diversification over hundreds of millions of years of geological time. Evolutionary time on a geological scale is the domain of paleontology, and calibration of rates for numerous important processes depends on evidence in the fossil record. The fossil record is necessarily the ultimate test of many systematic and evolutionary hypotheses. Primates and mammals in general have a relatively dense and continuous fossil record, permitting hypotheses of relationships and rates to be explored in more depth than would otherwise be possible. Different approaches to evolution and different scales of inquiry yield patterns appropriate for understanding different processes. Some approaches answer specific questions while others are more general. The genetic relationships among the living hominoids have been the subject of numerous molecular studies over the past 30 years. Recent DNA sequencing studies have focused upon resolving the genetic relationships among the major lineages of Old World monkeys. However, a complete evolutionary tree, or phylogeny, includes both the branching order and times of divergence of the species. Estimation of divergence dates within molecular phylogenies requires external calibration of the rate of molecular evolution, typically through interpretation of a few key fossils from the group of interest. From the early Catarrhine fossil record, the date of divergence of the Old World monkey and the hominoid lineages has often been estimated to be about 25 to 30 million years ago. This figure illustrates the primate phylogeny models. During the past 4 decades, the technology of determining molecular structure has advanced at a rapid pace and has become much more accessible. When the idea emerged that studying genetic material in biology is fundamental to the understanding of all animal life, it caused great excitement and enthusiasm. There can be no doubt that the subject of human molecular genetics provided deep insights into the way our body works. Scientists are now able to selectively isolate and study even single fragments of DNA thought to be genes. Now, within this framework was the recognition that genetic material also provides insights that makes it possible to reconstruct the evolutionary history of extant organisms, and application of this ability to the study of non-human primate began. Probably the first application to several primate species long before the 1960s was the serological research by Natal in 1904. Since its beginnings, molecular approaches to primatology have often been controversial. Researchers learned that it was not always possible to solve unequivocally many of the phylogenetic and taxonomic puzzles within the primate order with these new approaches. Now this figure illustrates the molecular phylogeny of living primates. DNA is the crucial coating substance that is replicated every time a cell divides. Similar to morphological studies, molecular studies require that recognized characters be compared with similar characters that are declared to be primitive or primary. Many molecular studies necessarily remain phenetic, which means based on similarity of phenotypic characters. It is impossible to be anything but phenetic for immunology and DNA hybridization, where there are no characters present, only distances between pairs of taxa. Thus, each evaluation is firmly based in other previously established assumptions about the rate of evolutionary advance of the species that are studied. Now let us discuss about the different methods to study molecular phylogeny. Precipitin reaction assay. One of the earliest indirect methods applied to primates and many other mammals in the attempt to decipher the evolutionary relationships is called the precipitin reaction assay. It makes use of the functions of the immune system and its reactions to foreign protein molecules. The preferred vector initially was blood serum. Basically, this analysis measures the degree of in vitro clotting or precipitate formation. First, antigens from the organism being tested are injected into a different animal, usually a rabbit or chicken. The resulting antiserum that builds up is mixed with either the original donor blood or the blood of closely related species. Application of this precipitate elicits different degrees of coagulation in the tested blood. Thus, for example, human antiserum not only causes a strong reaction with the donor human blood, but also with the blood of chimpanzees, gorillas, macaques, and so on in decreasing intensity according to taxonomic distance. Octolony immunodiffusion method. Now, another method of studying molecular relationship is to analyze antisera to blood plasma or serum, a method that was perfected by Octolony in 1958 and bears his name, Octolony immunodiffusion. Immunodiffusion uses two protein samples that are simultaneously diffused or sprayed against an antiserum or an agar gel-based plate. Initially, in investigations involving primates, antisera were made to plasma or serum. Later, this method was further developed and elaborated by making antisera to single purified proteins. This technique was used to test primate albumins, which are synthesized early during fetal development by Goodman in 1962, who simply confirmed primate relationships more or less as they had originally been established based on morphological similarities and dissimilarities. Immunodiffusion technology is still used in immunological comparisons of proteins for phylogenetic evaluation. Researchers continue to use immunodiffusion techniques to investigate the evolutionary relationships among primates. Unlike earlier studies, and because of deeper understanding about molecular structure and reactivity, the sera used in tests are now purified. The antigen-antibody reaction is proportional to the closeness of relationship between the primates tested. Molecular clocks. It soon became obvious that it is not possible to properly calibrate any biological event. Neither biology nor evolution ever follow a steady time frame. The characteristic of biology is that it is irregular and that it functions in a random pattern. Consequently, it is not possible to properly calibrate biological events or to formulate strict invariable biological laws. Bohr in 1974 studied individual purified proteins rather than non-purified proteins. He prepared antisera to a series of purified human serum proteins in rabbits and compared them with serum proteins of other primates. Bohr graded up to 15 antisera to different serum proteins and tested them against serum samples from 3 apes, that is, Ban, Gorilla, and Bongo, for Old World monkeys, Magaga, Cercopithecus, Erythropicepus, and Cercocepus, 1 New World monkey, that is, Kipus, and 1 prosimian primate, that is, the calico. The resulting evolutionary tree was in total agreement with the evolutionary tree created with the help of morphological characters, the calico on its own branch, as are the New World monkeys, the Old World monkeys on the third branch clustered with the 3 great apes, that is, the orangutan, gorilla, and chimpanzee, the gorilla and chimpanzee being closest to humans. Now let us see immunoelectrophoresis method. The next step forward in the investigation of protein was the application of electrophoresis, which studies the differences that relate to size and electric charge of protein molecules. Electrophoresis separates the charged protein or the nucleic acid molecules according to their neat electrical charge and mass by drawing them through a filter material, which may be ether, vapor, or gel, using an electrical field. The molecules migrate in narrow bands along wicks toward the electrical pole that is charged opposite to their own electrical charge, from one pole to the other at a differential rate. They can be stained to make them move visible and subsequently compare between species. Two-dimensional resolution of electrophoresis was applied to primate molecules with the help of starch gel electrophoresis and agar gel precipitin testing, examining the reaction of a variety of antisera to proteins. Now let us discuss about protein radioimmunoassay. Now today, radioimmunoassay is carried out when only minute amounts of protein are available for evaluation. This technique has also been applied to fossils. Evaluation requires the material to be radioactive. Albumin is often used for this procedure because it is abundant among vertebrates, relatively stable and easily purified, and requires only small amounts of tissue. Now let us see microcomplement fixation method. Serum albumins are predominantly used in an expeditious procedure that allows comparison of differences between homologous proteins. Microcomplement fixation uses reactions between soluble antigens and antibodies that are in a dilute solution. In this medium, only high-affinity antibodies will react with their antigens. Basically, a serum is tested for its reaction with an antiserum as it is progressively diluted. At each step, there is a point when maximum precipitation occurs. This method was applied to help determine the relationship between humans and chimpanzees by Serac and Wilson, as well as to the study of albumins and transference by Cronin and Serac to decipher the taxonomic relationships among Old World monkeys. Now let us discuss about major histocompatibility antigen recognition. Another procedure that measures variations among major histocompatibility antigens is used in molecular primatology studies. These glycogenes are involved in antigen recognition when an immune response to some foreign substance occurs. Major histocompatibility antigens vary between individuals. Two classes of these antigens are recognized as 1 MHC antigens and 2 MHC antigens. Class 1 MHC molecules are triggered by antigens that originate within the cell and are therefore called endogenous antigens. Fragments inside the cell that originate from a foreign protein, such as a protein encoded by the genes of a virus, become bound to the 1 MHC molecules that, unlike class 2 MHC cells, occur in almost all cells with a nucleus. Class 2 MHC molecules are found only on the surface of cells that are involved in immune reactions. They are therefore called exogenous antigens. Class 2 MHC molecules are located on macrophages that process foreign antigen fragments on the outside of the cell. Many researchers have studied hemoglobins, types of proteins or amino acid sequences, as part of their endeavor to solve questions in primate phylogeny. One result from hemoglobin research that was carried out in the 1970s was the obvious conclusion that the loricid Nycticebus is not monophyletic with the lemur genera Eulemur and Propithecus, but belongs to a branch between lemurs and anthropoid primates. Now let us discuss about the direct study of genetic material. In contrast, techniques that allow the direct analysis of DNA have become more widely used during recent years, and they have been applied to problems of population genetics as well as systematics. Importantly, direct phenotype studies are now applied in the assessment of genetic diversity of endangered animal populations, which is crucial for planning species conservation in the future. Such analyses are also advantageous because small tissue samples are sufficient for analysis and DNA can even be obtained from extinct taxa. And these techniques are as under, we have DNA hybridization and polymerase chain reaction. One of the techniques that allow direct comparison of different DNAs is the DNA hybridization technique. This method makes it possible to estimate the degree of DNA sequence differences between the genomes. DNA hybridization is a potential tool to study DNA sequence evolution that supplements phenotypical evaluation of morphological characters. Therefore, since these methods of deciphering evolutionary process are independent, when the results of both are combined and evaluated together, they can potentially be of great importance for the understanding of evolutionary sequences. The techniques are as under: restriction fragment length polymorphism analysis. Another way to study DNA directly is DNA restriction analysis and restriction fragment length polymorphism, which identifies DNA pieces and the internal structure with the help of restriction enzymes. Genomic DNA can be fragmented or cut into smaller pieces by bacterial or yeast enzymes called as restriction endonucleases. Most restriction endonucleases work by recognizing a specific 6-base bare palindrome sequence that is reading exactly the same from left to right and right to left and cutting the DNA at these sequences. The results are fragments of DNA of varying length that can be separated and visualized with the help of electrophoresis. This figure illustrates the evolution of microRNA in primates. Now let us discuss about the Southern blotting method. One very useful technique that makes it possible to study single DNA genes was described by Southern in 1975 and became widely known as the Southern hybridization or Southern blotting. Certain separated RE-treated DNA fragments with the help of electrophoresis on an aqueous shell strip, identification and separation of single DNA sequence is thus made possible as the radioactively marked probe and the gene to be studied are made visible by autoradiography. For example, the taxonomic position of the enigmatic prosimian genus Tarsius in relation to other primates has been re-evaluated by Coop et al. In 1989. Coop and his colleagues concluded from their investigation that beta-globin sequences, globins are the protein constituents of hemoglobin. Each hemoglobin molecule consists of 4 globin subunits. They then opine that it is justified to place Tarsius together with anthropoids in suborder Haplorhini. This placement, however, is solely based on the evaluation of findings regarding one globin gene cluster, that is the beta-globin cluster, and a single globin gene, which is just a globin, a gene from the A globin gene cluster. Now this figure illustrates the molecular phylogeny and evolution of prosimians based on complete sequences of mitochondrial DNAs. Mitochondrial DNA has been extensively studied in the genus Macaque, a geographically widely dispersed and species-rich group of old-world monkey. All the studies have been done under the assumption that mitochondrial DNA is totally lost from the paternal line and is only maternally inherited. Human mitochondrial DNA has also been used to investigate migration patterns of human populations. For example, it has been shown that the people of Polynesian islands are genetically homogeneous and their mitochondrial DNA is very uniform. The Polynesian islands are therefore thought to have been only very recently colonized by a small human founder group. In contrast, the human population of the Western Pacific Islands shows higher levels of genetic diversity. Therefore, presumably, they were settled much earlier in human history than Polynesia. Now let us discuss some of the problems of phylogenetic analysis using molecular data. In primatology, molecular data have no real meaning unless they are brought into a phylogenetic context. Although there have been many attempts to give phylogenetic significance to findings about proteins, immunological differences, DNA sequences, or mitochondrial DNA correlations, these have often been contradictory and disappointing. The reason for this lies in the functional background of molecular data. Even if molecular differences can be documented, they usually cannot directly be correlated with any morphological features. Such undesignated characters must then be analyzed. An additional problem is the fact that most of the genetic coding regions called exons are separated by extensive areas of non-coding regions, which are known as introns. This simply means that most of the DNA in eukaryotic cells is not made up of functional genes. More questions than answers are resulting from the multitude of new research. One serious problem remains the lack of communication between research groups using different methodologies. Now let us summarize this module. All the methods of molecular study that we have discussed permit only indirect conclusions about the fundamental genetic basis underlying the structure of the protein studied. Hence, there are procedures that understandably open up numerous inroads for inaccuracy. Molecular primatology is especially useful in long-term research of both population dynamics and population genetics among wild primate populations in the attempt to decipher how evolution proceeds. DNA molecules can be helpful in solving uncertain paternity relationships among individual primates within groups that are the subject of long-term social behavioral studies. In addition, genetic fingerprinting is an indispensable tool for preserving genetic diversity of captive primate populations as part of the attempt to conserve and eventually reintroduce back to the wild endangered primates. Evolutionary or taxonomic evaluation of molecular data is only practicable when it is rooted in conventional morphological assessments. A multitude of molecular studies address human biology in particular and primate biology in general. However, many of the studies use techniques that are not compatible with each other or are based on molecules of unknown function and hence are not always directly comparable or meaningful. Well, evolution is a complex subject encompassing the history of life and its governing processes. Our understanding of evolution is based on interpretation of patterns of diversity, ecology, behavior, visible morphology, and invisible molecular structure that have changed and continue to change through time. The genetic relationships among the living hominoids have been the subject of numerous molecular studies over the past 30 years. Recent DNA sequencing studies have focused upon resolving the genetic relationships among the major lineages of Old World monkeys. However, a complete evolutionary tree or phylogeny includes both the branching order and times of divergence of the species. DNA is the crucial coding substance that is replicated every time a cell divides. Similar molecular studies require that recognized characters be compared with similar characters that are declared to be primitive or primary. Many molecular studies necessarily remain phenetic, which means based on similarity of phenotypic characters. Now, it is impossible to be anything but phenetic for immunology and DNA hybridization, where there are no characters present, only distances between the bases of taxa. Methods to study molecular phylogeny been provided, such as octolony immunodiffusion, molecular clocks, immunoelectrophoresis, protein radioimmunoassay, microcomplement fixation, major histocompatibility antigen recognition, and mitochondrial DNA. Direct study of genetic material can be done through DNA hybridization and polymerase chain reaction, restriction fragment length polymorphism analysis, and the Southern plotting. That's all for this module. Thank you so much for listening.